Hi, I'm Katie Totten, and I have done prompt 41B, different genetic mutations. The first type of mutation we will look at today is missense. Missense is a single point mutation in which a single nucleotide changes, resulting in a different amino acid. The top strand seen here is the DNA sequence that we will be comparing to the mutant strand to illustrate these mutations. The middle strand is the mRNA and the bottom is the amino acids. Now let's illustrate this by looking at mutation strand 1. By looking at the mutation strand on the bottom and comparing it to the DNA sequence on the top, where is the mutation? This mutation is found in the fourth quadrant of this DNA sequence between the C and the G. Changing the C to a G changes the amino acid. Before it was alanine and now it is glycine. This is a different amino acid which resembles the missense mutation. Switching between the C and a G is known as a transversion, which is going from a purine to a pyrimidine, or from a pyrimidine to a purine. The next mutation we will talk about is a nonsense mutation. This is when a single point mutation turns an amino acid into a stop codon. Now let's look at mutation strand 2. By comparing the DNA sequence on the top to the mutation strand, can you tell where the mutation is? This mutation is found in the second quadrant of the DNA strand, where a G is turned into an A. This now codes for a stop codon. Going from this G to an A or an A to a G is known as a transition. The third mutation we will talk about is a silent mutation. This is when a single point mutation does not change the amino acid that came before it. Now let's look at this with mutation strand 3. By comparing the DNA strand to the mutant strand, can you tell where the mutation is? This mutation is seen in the third quadrant of this DNA sequence, where the C was turned into a T. This ultimately does not change the amino acid that was coded for, illustrating a silent mutation. The changing of a C to a T is known as a transition. The fourth and final mutation we will talk about is a read-through. This is when a single point mutation turns a stop codon into an amino acid. Now let's illustrate this with mutation 4 strand. Compare the DNA strand to the mutant strand. Do you know where the mutation is? The mutation is seen in the sixth quadrant of this DNA strand, where the G is turned into a C. This turns a stop codon into an amino acid, resulting in a read-through mutation. The G being replaced with a C is known as a transversion. Thank you for watching my video.